Hello and welcome back. Today we're going to talk about cell structure and function. As you very well know, cells are really super small. Maybe you've never thought about why there aren't giant cells in the world. But if you take a look at this graphic here, let me get my pen up here in writing. So you can see that cells are really small, and humans are not unicellular. We're not made of one cell, but more like we're like multicellular, and we're made of trillions of cells. So why are cells not gigantic? Well, it all goes down kind of to the mathematics of it. So if you look at this small cell right here, if you were to do the total surface area with this arbitrary unit using 1, surface area being height times width times number of boxes times number of sides, you get a calculation of 6. But if you calculate its volume, which is length times width times height times number of boxes, it's only 1. So when you take 6 and do the surface area to volume ratio, 6 divided by 1, that gives you a total surface area to volume ratio of 6, which is pretty high. Now if cells were huge, so if you took and said that the side was arbitrarily 5, if cells were huge, you can see that the total surface area is 150, total volume is calculated at 125, and if you did the surface area to volume ratio, you get basically 1.2. And that's much smaller than the unit of 6. So what that's saying is there is a huge amount of volume compared to the surface area. Okay, so you can see here that there's a huge amount of surface area compared to the little bit of volume. Now, if we take a look at multicellular organisms like yourself, we have huge amounts of volume, but we have huge amounts of surface area. And so when you look at the surface area compared to the volume, you get that calculation of 6, just like a small cell right over here. So what's the advantage of having a large surface area to volume ratio? We know that there's a lot of various reasons why that's really super important. I guess I forgot to tell you that this just by definition, a cell is the smallest unit of life that has the ability to reproduce, metabolize, move, adapt, and create waste, and that's what we're created of, is huge numbers of cells. But going back to my original thought there, why are cells small? Well, cells are small because it gives them an advantage. So the advantages are tremendous. One of them is that cells, because they're small, have fast rates of diffusion, that is the movement of molecules across the membrane. So cells are faster at doing osmosis, which is the movement of water into and out of the cell. All different kinds of transport of substances is much faster, such as the absorption of nutrients, removal of waste from cells, because of that high surface area volume ratio. So we're able to get rid of heat very efficiently. If cells were gigantic, they would basically fill full of heat and perhaps even destroy themselves because of the heat that's being formed. They're able to produce enough energy to run the cell. So if you had huge amounts of volume, maybe you wouldn't be able to provide enough energy for the cell or get enough of the materials into the cell that you would actually need. And getting materials and resources in, it happens because of that large surface area to volume ratio, and getting waste products out all occur because of that large surface area to volume ratio. So you can see surface area to volume ratio is a really super important part of being a cell and why cells are small. So if we move on down to talking about cell parts now, we are eukaryotic, which means we have a nucleus. And so I want to talk about the general animal cell during the course of this lecture. So we're going to go through and we're going to look at ribosomes, the Golgi apparatus, the cell membrane, the mitochondria, the lysosome. We'll talk about flagella, we'll talk about cilia, and we'll talk about microvilli and peroxisomes. So, and of course, smooth and rough ER. So these are some of the parts that we're going to talk about. I'll take each part and talk to you about them for 30 seconds to a minute. Now, organelles are small organ-like structures. I put organs in quotation marks. Organ-like structures in cells that have very specific jobs that they perform. The nucleus is the first part I want to talk about, and it has a really critical job. So the job of the nucleus is to house most of the organism's DNA or genetic material. And I say most because not all of it's in the nucleus. Your genes are coded instructions to build all the things the cell needs. So all of that is housed inside of the nucleus, and there's a lot of genetic material that's inside of that cell part. Another job of the nucleus is to synthesize, which means to make and assemble these structures that are called ribosomes. So the DNA has coded instructions that actually build ribosomal RNA, which is the molecule that actually makes up the ribosomes. So ribosomes, we'll talk about in a few minutes, and they're just another cell part that is very important to the function of the cell. Another job of the nucleus is to synthesize or to make messenger RNA. We also term this mRNA, and these are copies of the blueprints to make products for the cell. The DNA never leaves the nucleus except during cell division, so we have to make copies of the blueprints of the genetic material and then send those blueprints out specifically to the ribosome in order to be made into a product such as a protein. And the structure of the nucleus is that it's double membrane bound, so it actually has two membranes. So if you were to look at a nucleus, it actually has two membranes. And these membranes are phospholipid bilayers. Okay, so they're very complex phospholipid bilayers. That's what each of those membranes actually looks like. Okay, and there are pores or holes through them, as I'll show you in the next graphic. So they are porous. So here's a picture of a nucleus, and you can see the little nuclear pores there. It is double membrane bound. You have one membrane here and you have one membrane over here. But there are pores that span throughout the nucleus. You can see those pores from the front surface there. This is kind of what they look like as they're going through the membrane. So substances can come into and out of the nucleus because of those pores. So this organization of being double membrane bound and having pores helps to segregate materials that are inside the nucleus from materials on the outside of the nucleus. So it creates a boundary. Messenger RNA, ribosomal RNA, building materials, proteins, ATP, which is an energy source, all of those things can travel into and out of those pores. But not everything can move through those pores. DNA doesn't move through those pores, so they are selective about what comes into and out of the nucleus. And they're trying to protect the nucleus from various things and protect the DNA from enzymes that are on the outside surface of the cell. And that genetic material that's inside of the nucleus in a non-dividing cell, typically it's all spread out, and it's in a form we call chromatin. And you can see that the DNA is here. It's made of those building blocks we talked about before, the nucleic acids. And I can see the little nitrogenous bases here in the rungs of the DNA ladder. That DNA is organized. It actually is coiled around nucleosomes, which are little proteins that help to organize the DNA. Histone proteins help to basically take these nucleosome beads and connect them together to compact the DNA, and you can see the DNA is becoming compacted in this little model over here. When a cell goes through cell division, the DNA is supercoiled, that is, all the DNA is packed and supercoiled and organized into the structure that we actually call the visible chromosome. And so that it helps it to be organized so that when you go through cell division, that the chromosomes can be equally separated into different copies, which can go to different sides of the dividing cell. Okay, so this is what it typically looks like in a non-dividing cell. This is what it looks like in a dividing cell in the form of a visible chromosome. So we have these nucleosomes and these histones. They help to compact the DNA down so it actually fits inside of the nucleus and is organized inside of the nucleus. All right, well, moving away from the nucleus, we'll go ahead and do the ribosomes. The ribosomes, they have the job of reading the messenger RNA or the blueprints that came from the nucleus and make proteins from the reading of those blueprints. The structure, it's composed of ribosomal RNA that comes from the nucleus, and it's also composed of proteins. These proteins and ribonucleic acid come together to form the structure of the ribosomes. they're not surrounded by a membrane, but rather they're made of two subunits. They actually have a large subunit and a small subunit, and these are actually separated in a cell that's not that's not reading messenger RNA. When messenger RNA actually comes in, these subunits come together, and the messenger RNA will actually be read by the two subunits that are together. Okay. Sometimes we have free ribosomes. These are ribosomes that are just in the cytosol. And sometimes we have bound ribosomes, and these ribosomes are attached to membranes like the rough endoplasmic reticulum that we'll talk about in just a few minutes. So bound ribosomes are attached to the nuclear membrane or the endoplasmic reticulum, and they're going to make proteins very specifically that are going to be inserted into membranes, your cell membrane, or exported outside of the cell, or packaged into these little vesicles called lysosomes. Lysosomes contain very powerful enzymes that we use to digest products. Free ribosomes, on the other hand, are going to be suspended in a cytosol. They're not attached to a membrane, and they make proteins that function as enzymes in the cytosol. Okay, that's the liquid portion of the cell. Now, one oddity, or one cool thing, is that bacterial ribosomes are smaller than eukaryotic ribosomes. And because they're smaller than eukaryotic ribosomes, we can use antibiotics to inactivate those particular ribosomes. So if you've ever taken an antibiotic before, if that antibiotic was something like tetracycline or streptomycin, these particular antibiotics actually knock out bacterial ribosomes, but they don't take out your ribosomes. There are antibiotics that actually deactivate our ribosomes, and of course we wouldn't want to eat those because it would kill you. When you deactivate or inactivate bacterial ribosomes, they can't make proteins, and if they can't make proteins, they can't grow, they can't reproduce, and they can't repair themselves, and they're going to die within days. Well, within the cells, there is an endomembrane system, and what this is a series of membranes that will transport substances between these various components. So the smooth ER, the rough ER, the Golgi apparatus, these all are membranes that are going to communicate with each other and send products that they make to each other, and this is how things are made inside the cell and then transported to the outside cell and dumped outside. Okay, this is how things are made inside the cell and kept in things like lysosomes. So let's go ahead and we'll talk about the components of the system. The first component of this sophisticated system is called the endoplasmic reticulum. This is a network of tubules, which are small tubes and little sacs, little containers that house various things that the endoplasmic reticulum can make. So the endoplasmic reticulum is a series of membranes that make internal boundaries or compartments, and there's two types of them, rough ER and smooth ER. The rough endoplasmic reticulum has many ribosomes that are attached to its membrane. Now you know that ribosomes are little protein factories. They're going to make proteins based on the messenger RNA message coming from the DNA. It's involved in making proteins that will be secreted, that is, taken to the outside of the cell and dumped out, attached to the membrane, or for use in the cytosol, for example, making proteins that will be put into lysosomes. They actively make more membrane as well. So your cell membrane wears out, and this is the part of the cell that actually makes more membrane and helps to repair membrane that's or to replace membrane that's been damaged or destroyed. And this is what it looks like under an electron micrograph. So if you take an electron microscope and take a picture of it, this is kind of what it looks like. And you can see the little dots inside are the ribosomes, and the little lines that you see are the actual membranes. This is an artist's three-dimensional rendition of it. The next of the endoplasmic reticulum is the smooth endoplasmic reticulum, and they have no bound ribosomes, so that's why they are considered smooth. They look smooth when you look at them through the electron microscope, and they have many jobs they perform. These are really important parts. One of the jobs is to synthesize various kinds of lipids. They synthesize phospholipids, which can make more cell membrane. They synthesize things like steroids, which would be like testosterone and estrogen and cortisol. They do make cholesterol, which are the building blocks of steroid and steroid-derived products. And they do make triglycerides, okay, which are your, like monoglycerides, diglycerides, and triglycerides. They also synthesize, that is, they make and they store glycogen. Okay, glycogen is made from glucose monomers that are stuck together to form the polymer glycogen. And so they make them, they store them, and they can release them. So the liver and muscle are two organs that you would see a lot of. They would have cells that would contain lots of smooth ER to do this particular job of making glycogen. They also absorb, store, and release calcium ions. This is very important in things like muscle tissue. Another thing that you see in liver cells, but there are in other cells as well, is the smooth ER can detoxify or inactivate drugs and poisons that we ingest. So the liver cells, because of the way we digest food, the liver is the first organ that receives nutrients from the small intestine. So literally all the blood that's absorbing nutrients from the small intestine goes straight to the liver, and it's filtered by the liver. And the liver cells help to, basically because they have lots of smooth ER, they help to detoxify drugs and poisons that we ingest in our food that we eat. So if you're exposed to poisons, alcohols or poisons or drugs, the smooth ER in the liver will help to detoxify that. And if it escapes through the liver, then it comes back because the blood always goes to the liver, and constantly the liver is taking alcohol and poisons out of your circulation. Now the liver, because of your exposure or repeated exposure to poisons or alcohol, is responsible for tolerance of drugs. So, people that take drugs have to take more drugs each time they take drugs to get the same effect, because their liver cells are literally adapting to the drugs they're taking, and they're making more and more smooth ER. If you make more smooth ER, then it will take drugs and alcohol out of your system much more quickly. So you want to be real careful that you don't take drugs and alcohol, so that when you do have to have things like anesthesia or other kinds of drugs to cure you from a disease, that you don't have too much smooth ER that's taking those drugs out of circulation. Well, the Golgi apparatus is our next part of the endomembrane system, and what it's going to do is it's going to modify, store, sort, and ship any kind of product that's coming from the endoplasmic reticulum. And its structure is that it consists of a series of stacks of flattened membranes, and it has two faces. It has what we call a forming face, and it has an eroding face. Let me kind of show you what that kind of looks like. So if this were the endoplasmic reticulum over here, and it's basically taken and making proteins, it's going to send those proteins to the side of the Golgi apparatus that is called the cis face, or the forming face. So these little vesicles that it makes, it actually transports those vesicles with the proteins inside. It transports those to the Golgi apparatus. Those little vesicles will fuse with the membranes of the Golgi apparatus, and inside of the Golgi apparatus, very complex things occur. That's why your modification, your sorting, and then eventually your shipping actually all occurs inside of those membranes. The trans face over here is the shipping side, or the eroding side, and so you get little vesicles that erode off of that, so it keeps the proteins inside of those little vesicles separated from the rest of the cytoplasm. And those little vesicles, they can go on to make lysosomes, they can go on to dump the products to the outside of the cell. So for example, if you have a cold, and one thing that you do when you have a cold is your mucous membranes produce lots of mucus. So we have, inside of the nucleus, we have DNA, which codes for the making of the mucus proteins. Those codes come out in the form of messenger RNA. They go to the ribosomes of the endoplasmic reticulum, and they make those little proteins that are mucus. Those little mucus proteins will be transported to the Golgi apparatus, and eventually they will be eroded from the Golgi apparatus into little vesicles. Those little vesicles will carry the little mucus proteins, and they'll dump outside, and of course you have to then blow your nose. Okay, so that's just an example of how, we make a protein, transport it to the Golgi apparatus, and then dump it to the outside of the body. This is what it looks like under electron microscopy. It's a really neat looking little organelle. I like to think of it as like a stack of pancakes, and I also like to think of the Golgi apparatus as being like the UPS for the cell. UPS ships, sorts, and packages proteins or packages for us, and then the Golgi apparatus does it for us in terms of making protein packages. So there's all kinds of markers on the surface of these vesicles. That's how the cell knows how to keep them inside the cytosol or dump them to the outside of the body. So markers on the surface are that would be just like packing slips that we have, and it aids in the sorting and the sending of the vesicles to various parts of the cells. So there is a barcoding effect. There are little protein barcodes on the surface of these vesicles, so the cell knows exactly what to do with all of these particular products. It doesn't get confused. So the Golgi apparatus came like a lysosome. The job of the lysosome is to contain hydrolytic enzymes. Hydro means water, lytic means to destroy. So these enzymes use water to destroy or to break down various kinds of macromolecules. It can break down proteins using proteases. It can break down nucleic acids using nucleases. And it can break down various kinds of polysaccharides and disaccharides using carbohydrases. These are all enzymes that are going to help to aid in digesting the food that the lysosome is digesting. Now, your immune system cells actually have lots of lysosomes, and they use lysosomes to kill pathogenic organisms that come into your body. We also use lysosomes for autophagy. Autophagy is a concept where we take and eat worn out body parts or put them into vacuoles, and then we take lysosomes and recycle them using the digestive enzymes that are inside of them. So they are membrane-bound sacs that are created by the Golgi apparatus, and they only contain one membrane. So they are a boundary. Remember, the hallmark of a living thing is having boundaries. So the lysosome is a boundary. Now, why do you want to contain the enzymes inside of a boundary? Well, you want those enzymes to be inside of that structure so that they don't go out and destroy cell parts. So the pH inside of a lysosome is very low. There are little proton ports that take hydrogen ions from the outside and pump them to the inside, and that decreases the pH of the interior of the lysosome. And that's a protective mechanism in case of leakage. So if all of a sudden this lysosome leaked all of its enzymes to the outside, these enzymes only work at a low pH. So the cytosol is at a higher pH, so it actually deactivates the enzymes from working. So that's kind of a cool little protective function of that. This is just showing you some of the concepts we just talked about. So the messenger RNA carries blueprints to the ribosomes of the endoplasmic reticulum. The rough endoplasmic reticulum makes those proteins, transports them to the Golgi apparatus. Sometimes the Golgi apparatus will make transport vesicles that will transport things to the outside of the cell. Sometimes they make things like lysosomes, which keep the proteins inside the cell. Here, if you eat a little piece of food, you can see a food vacuole forming. The lysosome comes down, joins with the food vacuole, and digests the food molecules, and those food molecules can be released to the cytosol. In autophagy, a worn-out cell part is contained inside of this little vacuole. A lysosome comes down, attaches to it, and then we have autophagy, which is the digestion of worn-out cell parts. And that's a good thing. You don't want the worn-out cell parts to accumulate inside of your cell. So it's a way we can recycle and digest nutrients and give those back to the cell to use. That's called the endomembrane system. It's very sophisticated. One day, if you continue in your education, you'll take a cell biology course or a molecular biology course, and they'll go into great levels of detail about how that's organized. Well, vacuoles are structures that are membrane sacs. They only contain one membrane, and they're a little bit larger than smaller vesicles. Their job is to contain food or to contain worn-out cell parts inside of our cells. Food vacuoles form during phagocytosis. So if you have a food particle there, You can see the food particle, the cell will actually wrap its membrane around it and contain it inside of a membrane. So that's a food vacuole. And we do that during this process called phagocytosis. Phagocytosis is also a way that your immune system cells will take things like bad bacteria, engulf those bad bacteria, and contain the bad bacteria inside of themselves, and then they'll use, lysosomes will come over and attach to this and help to kill the little microorganisms that's inside that vacuole. So we use these as an important defense against invading organisms for sure. The mitochondria basically are contained by or enclosed by two membranes, so they're double membrane bound. And if I just drew that structure for you, so it basically has two membranes, two phospholipid bilayers. And we think that is true because it's an endosymbiont. We think that in the past, a long time ago, your ancestral cells actually took and ate a bacterium and retained that bacterium inside of our cells. It actually, instead of being eaten, became part of the cell and helped to function, it helped the cell to basically generate ATP or energy. Now, there's a lot of lines of evidence to suggest that that's an endosymbiont. Mitochondria contain their own DNA. They are the size of bacteria. They have a chromosome that is bacterial. It's a circular chromosome. They actually have bacterial genes. So there's a lot of lines of evidence to suggest that that's actually true, that they actually are little organisms that live inside of each one of our cells. So, you're never truly alone. Every one of your cells has little creatures living inside of it called mitochondria. So, the mitochondria contains mitochondrial DNA, and what's kind of cool about mitochondrial DNA is that you inherit all your mitochondrial DNA from your mother. Your dad doesn't contribute mitochondria to you, so all your mitochondrial DNA is contained from, or you acquire from your mother. And that's kind of interesting because you can actually track your maternal lineage by looking at mitochondrial DNA and analyzing mitochondrial DNA. You can, so if ancestry is important to you and you want to know where your mom came from, look at your DNA, get it analyzed, and it'll tell you where your mom's people came from. The mitochondria houses many, many, many enzymes that are going to do jobs for us. The job of the mitochondria is cellular respiration. So it's the breaking down of the food that we eat to make ATP, to make energy, ATP. That's a pretty sophisticated process. It's not one I'm going to talk to you about now, but we might discuss it a little bit when we do muscle cell physiology. So you can see the outer membrane, you can see the inner membrane. So there is a matrix inside, kind of a cytosol inside, and it's highly folded. The inner membrane is highly folded to increase the surface area at which cellular respiration can occur. So that's the mitochondrion for you. The next cell part are peroxisomes. Peroxisomes function to break down fatty acids that come inside of our cells. Fatty acids are very common. In the process of breaking down these fatty acids, the peroxisome makes lots of hydrogen peroxide, which is pretty toxic to the cell. Luckily, though, we have an enzyme called catalase that takes the hydrogen peroxide and breaks it down into oxygen and water. If you ever want to see that process take place, take some hydrogen peroxide, a very small percentage of it, like you have at home, which is probably a 2% solution. Take that and put it into your mouth and swish it around. What you'll see or experience is bubbling that will occur inside your mouth because there's catalase inside your mouth. And the bubbles are oxygen bubbles, and it creates oxygen bubbles in water. So we use hydrogen peroxide as a disinfectant. Your cells use it too. There are cells in your body that produce hydrogen peroxide to actually destroy microorganisms, but the peroxisome uses it to it's a product that basically comes from the breakdown of fatty acids. Another job of peroxisomes is to detoxify alcohol and other harmful substances. So it has chemicals inside of it that help them to do that particular job. It's only one membrane bound, so essentially it has one membrane. And you can see that it looks like this. It's very simple in structure. You have a membrane around it, and inside of it you can see there's just a little crystal inside of it. So it's kind of a cool looking structure. Well, the cytoskeleton functions to support the cell shape. It provides anchorage for the organelles and for enzymes. So there, everything is held in place by the cytoskeleton. I like to think of it as like the little bones and muscles of the cell to provide support and move things around. It does provide for motility. There are little motor molecules that are associated with the cytoskeleton to help move little parts around. So here is a microtubule that's part of the cytoskeleton. There are also microfilaments we'll see in a minute. But you can see the microtubule serves as the skeleton of the cell, and these little motor molecules under the energy source ATP can actually move organelles from one place or one side of the cell to another. The little motor molecules can actually cause microtubules to slide past one another. They can cause motility when you talk about parts like the flagella or the cilia that we'll look at in a minute. Another job of the cytoskeleton is to provide for phagocytosis. And you can see that these little microorganisms are the cytocell membrane is reaching out and grabbing these little microorganisms out here, and it's bringing them in and eating them. And that's a process called phagocytosis. So the deformation of that of the shape of the membrane, you can see it's extending out. Those little deformations of the cell membrane are because of the cytoskeleton allowing them to actually do that. So this is what the cytoskeleton looks like if you were to take a picture of the interior of the cell. The interior of the cell is not just a bunch of liquid, but it actually has great structure to it. You can see that there are some microtubules, which are more of the skeleton-like parts, and then there are some that are more muscle-like parts that are called microfilaments, and they're a little bit thinner than the microtubules. The microfilaments can contract and cause they can shorten themselves and cause movement of things. The microtubules are more like girders. They're more like, the studs inside of your house to create structural support. This is just showing you some of the movement of things. The mitochondria can move because of the extension of the cytoskeleton out. Vesicles move because they actually are attached to the cytoskeleton and they're being transported. So all of your cell parts are moving around on these little monorails called microtubules, which are part of the cytoskeleton. So we do have a way to move and to organize and keep everything in its place. A very important part in the human body, and especially like things like intestinal cells and cells like inside your kidneys, is this cell part called the microvilli. Microvilli are where the cell membrane has folded to increase the surface area so that certain things can happen, like absorption of materials. So this is a microvillus. You can see it's an extension of the cell membrane, and inside of it there are all kinds of microfilaments or parts of the cytoskeleton that organize these little extensions to actually occur. You can see they're anchored down here into a web of the cytoskeleton, so it's a highly organized structure, but it increases the cell membrane surface area significantly for parts like kidney cells and intestinal cells, like small intestinal cells, to help to absorb things. Now your cilia are short hair-like extensions of the cell surface that are on the cell surface, and they're very numerous, and they provide for oar-like motion. You find cilia in places of your body like your trachea and your fallopian tubes if you're a female. So in those places the cilia help to move things. In the trachea the cilia help to move mucus, and the fallopian tubes the cilia help to move the egg cells from the ovary to the uterus. So several places, the fallopian tubes and the trachea would be places where you'd see cilia. Now, if you smoke, you kill your cilia, and that's why you have to cough to get mucus out of your trachea. So you've heard the smoker's cough before, I'm sure, because their cilia are dead. The flagella are long whip-like extensions of the surface of the cell, and they're few, and we really only have one place that I know of we have flagella, and that would be the sperm cell, and that whip-like undulating motions are going to cause it to actually provide for motion. So here would be a structure that has flagella, or a flagellum, which is the singular, and that causes the whip-like motion, the movement of the sperm cell. And this is an organism. This is like Peroxisome. It's an organism that has cilia all over the surface, but they provide oar-like motions. And again, we have those in several structures of our body, including the trachea and the uterine tube or fallopian tube. Okay, so I think I'll stop the video right here and just break this into two separate little videos. We'll do the cell membrane function and structure in our next little video. So until next time, I'll see you then.